Okay, so today we talked about cellular transport and just a quick review of the cell membrane, where we're going to be talking about molecules that are going to be moving from extracellular to intracellular. And then we're also going to mention about if something needs to move out from an intracellular to an extracellular range. So just a quick review of some terminology here. When we get to this word solution, a solution is made up of a solute and a solvent. So typically our solvent is water. Solutes could be things like sugar, potassium, sodium, calcium. The other three things to keep in mind as we talk about the four types of transport, there's three different characteristics: the concentration gradient, energy necessities, and carrier molecules. The concentration gradient, we use terminology up or down. Energy necessities, we use active or passive. And then carrier molecules, we use mediated or non-mediated. So when we talk about up or down, we're looking at the molecules, high concentration to low concentration. So these molecules, when we say down, we're going from high to low. When we're going from low to high, it's up or against the concentration gradient. Active or passive, active types of transport require energy in the form of ATP, and this is just a picture of the ATP molecule. Passive does not require energy. Mediated versus non-mediated. So is there help or is there no help? So both of these pictures are showing mediated methods of getting into the membrane that both involve a protein. The protein in this case looks like a channel. This one looks like a gate. So there's different proteins for different substances that these substances are going to recognize they belong. If we talk about non-mediated, meaning no help, it would be as if this molecule could go right through the membrane. So diffusion is our first type of cell transport. It's down the concentration gradient, passive and non-mediated. So if we take a look over here, it's going from high to low, and as it over time, you can see that the molecules reach an equilibrium. So there was no protein involved, there was no energy involved, they just simply diffused through the membrane. A good example of this is something like oxygen diffusing into the cell, carbon dioxide diffusing out. And those are small uncharged particles that can simply get through the membrane. This is happening all the time in our lungs and our respiratory system, and parts of the lungs called alveoli have lots of different surface areas so that this process can take place. Osmosis is the diffusion of water. So in this case, we need to work on some terminology. We have hypotonic, which means below strength, hypertonic, which means above strength. So hypo means below, hyper means above. And when we're talking about strength, we're talking about the amount of solute. Notice that the term hypotonic means below strength, so less solute, but more water. So I remember that hippos like to play in the water. So in a hypotonic solution, there's lots of water and less solute. Hypertonic, there's more solute, so that's these green, and there's less water. Now, the water is what's moving across the selectively permeable membrane because these large solutes cannot move across. So in a cell, water is going to move from hypo to a hypertonic situation. So this solution that this cell is in is hypotonic compared to inside of the cell. In this case, they're about equal. So we say it's an isotonic solution. It reached equilibrium, but the water molecules will continue to move back and forth. In a hypertonic solution, again, since these green solutes aren't going to move into the cell, water is going to leave the cell. This is important in real life. Real animal cells do not belong in a hypotonic solution because water might move in and the cell will burst or lyse. Homeostasis for animal cells is this isotonic condition where water is moving back and forth, kind of maintaining an equilibrium. If animal cells are placed in a hypertonic solution, water tends to leave the cell, and these cells will shrivel. They could clot, and forming clots can cause death as well. Now, homeostasis for a plant is turgid or this turgid or overwatered type of condition where saturating it with water causes the vacuole to be full and press against the cell wall. And that's the environment that a plant prefers for homeostasis. If some of the plant is in an isotonic solution where it's kind of balanced, we call that flaccid, and water is just sort of moving in and out equally. But if a plant is underwatered, it can risk the cell membrane being plasmolized. So you can see this green, this was the cell membrane right here. It's pulling away from the cell wall, and that could result in cell death. So if you want, take a look to see what happens when you put salt on a slug. You can look online to check that out and imagine what would happen if you put salt on a slug that has a pretty thin skin. Facilitated diffusion is the third type of transport. It's down the concentration gradient, passive, and mediated. So here we have molecules moving from high to low. That's why it's down. It's passive. There's no energy involved, but it does need a helper. Okay. So when we look at the cell membrane, these heads are polar. The tails are nonpolar, which means they don't like things that are charged or also polar molecules. So the particles that need help from a protein molecule to get through are large and charged molecules. Another name for a charged molecule is an ion, and that's because of the center region of the cell membrane. Now, a good time to pause the video and ask yourself is, what's the difference between a facilitated diffusion and regular diffusion? The answer is that facilitated diffusion is mediated. Regular diffusion is not mediated. Otherwise, they're the same. So the final type of cell transport is active transport. In this case, molecules are moving up the concentration gradient. So they're going from an area of low to high concentration, which is very different than what we've seen in the other three types. Active requires energy, so there's your ATP molecule being used, and it also requires a protein. So to learn more about a specific pump, read section 3.14 or 314 in your textbook, and it will explain a little bit more about how ATP helps that pump work. Some other types of active transport are endocytosis, and I'm going to click to the next, exocytosis. So they're pretty much opposites of each other, but sometimes the cell just needs to move large amounts of material, large particles, like maybe some giant polysaccharides, needs to move them into the cell all at once. So the particles kind of form a pit near this on the cell membrane, and then the cell membrane kind of closes off and forms a vesicle. If the cell is bringing in some particles, it's phagocytosis. If it's liquids, it's pinocytosis. So phagocytosis refers to cellular eating. Pinocytosis is cellular drinking. And knowing these prefixes are extremely important, like endo means in, cyto means cell. So again, phago means eating. You can think of food. Pino means drinking. And exocytosis is the reverse. So maybe the cell just made a bunch of proteins. Those proteins travel in a vesicle to the membrane, and then they're released all at once. I hope that is helpful. We then followed up with a packet in class to kind of review and apply the material that we just talked about today.